Hello and welcome everyone. In the last class, we discussed about epigenetic marks as well as what is chromatin and the types of chromatins. So, understanding the chromatin structure is very important for getting your hands on the basic mechanisms, the molecular mechanisms in genetics. So, I discussed that there are two types of chromatin, euchromatin and heterochromatin. Heterochromatin is transcriptionally inactive while euchromatin is transcriptionally active. So, heterochromatin is again of two types, constitutive heterochromatin and facultative heterochromatin. Sometimes, okay, so uh, sometimes some genes are required in certain cell types only. So, such genes are silenced in other cells. Why? Because obviously we don't need that gene, so we are not going to spend our energy on transcribing that gene. So, for example, is dopamine. This gene, this dopamine is a neurotransmitter. So, the gene producing dopamine neurotransmitter, it is required in neurons, right? But it is not required by muscle cells. So this gene will be active in neurons and it won't be, it will be silenced in muscle cells. So for silencing in muscle cell, it is heterochromatinized. Okay. It is in the form of heterochromatin in the muscle cells, while it is in the form of euchromatin in the neuronal cells, right? So such type of genes, such type of chromatin is known as facultative heterochromatin. This is known as facultative heterochromatin because such heterochromatin regions can be heterochromatin in certain cell types while it can be euchromatin in other cell types. Okay. While the other example of the heterochromatin are telomers and centromeres. So the central repeat regions and the repeat regions on the telomers, those repeat regions are heterochromatin. Okay, so such heterochromatins are so those regions are very important for maintaining the structural integrity of the chromosome. So like the structural integrity should be maintained in all cell types, and that's why those are heterochromatin in the form of heterochromatin in all cell types. So the example centromere and telomere as well as satellite DNA, all those type of heterochromatin are constitutive heterochromatin, means they are always in the heterochromatin stage in all cell differentiation stages and in all cell types. Okay, again, now when I told you that certain genes are silenced in certain cell types while they are required in other cell types, so there's another interesting example of the facultative heterochromatin is bar body. Okay. Now I'll be explaining you a phenomena called dosage compensation. This phenomena occurs to balance the ratio of sex chromosome to autosomes. So females in humans have XX chromosome while males have XY sex chromosomes. So they have only one X chromosome while the females have two X chromosomes. So balancing those two X chromosomes balancing the X chromosome to autosome ratio, what do? We inactivate one X chromosomes. So there will be inactivation of X chromosome. So this inactivated X chromosomes form bar body. I hope you got it. Alright. So these bar bodies are present in females. So this is type of facultative heterochromatin because those bar bodies are, if they in the females there are embryonic stem cells. In those embryonic stem cells, those both of the X chromosomes are active. But when those cells differentiates and they form adult cells, one X gets activated. So when it so this for getting inactivated, this X chromosome undergoes heterochromatinization, means conversion of euchromatin into heterochromatin. So at certain cell stage, at a certain cell differentiation stage, this X chromosome was actually euchromatin, and now it is turning to heterochromatin. Means it wasn't always heterochromatin form. It wasn't always transcriptionally silent. It produced some mRNA and then got heterochromatinized, and that's why this the bar body, comes into the facultative heterochromatin. Okay, I hope this is clear to you all. I forgot to discuss this in the last video. So let's start today's topic. It is DNA packaging. So now as we discussed in the last class, DNA is not as it is in the cell. It's not loosely present in the cell. It requires proteins which are known as histones. These histones pack the DNA in the cell nucleus. Alright, so these histones, when they pack DNA in the nucleus, they can pack those DNA structures at different levels of compactions. Now, why is it so? Why DNA needs to be compact at different levels of compaction? See, uh, each cell has around 2 meter of DNA. And what is the dimension of the cell? Its cell nucleus dimension is 6 micrometer. So in such small place you have to put 2 meter long DNA. Then you, what option you get? You have to wound it and put it in the cell nucleus, right? Imagine there's a lot of there's a thread kept, and if you wound this thread turn by turn in coordinated manner, you can put it in a small space, right? It's just the same. So the DNA is wound around histones, around the proteins, and it is kept in the nucleus. But this DNA also needs to be transcribed, right? And for transcription of the DNA to RNA, you need to have this DNA loose. Why? Because you have transcriptional machinery, and that transcriptional machinery is going to read the DNA. And when it is going to read the DNA, it will need the DNA loose, right? So that's why you have to make the DNA loose at some point of the cell cycle. And that is why at different stages of life cycle, the DNA in the cell is present in the different forms. For example, in interphase is metabolically active phase of cell life, and that's why the DNA is present in chromatin form. You know, this is the beads on the string structure. So the DNA is present in the chromatin form. Why? Because in the chromatin form, all of the transcriptional machinery can access the DNA, and it can then access the DNA, and it can transcribe it. And again, when from the interphase, from G1 phase, it will go to S phase, replication phase. The replication machinery will be able to access DNA, and DNA will be replicated. And that's why the chromatin structure, in the chromatin structure, all of the proteins, all of the machinery are able to access the DNA. And that is why in the metabolically active phase, means interphase, DNA is present in the chromatin form. In the prophase, it is still present in chromatin form, but when the cell starts to divide, it needs to pack the DNA into chromosomal form. Okay, so it packs DNA into chromosome form. Chromosome is the highest degree of compaction of DNA. That is why DNA is packed differently in different cell types. DNA package, why? Because DNA packaging is related to functioning. As I already discussed, if the DNA is compactly packed, like in the chromosome, it can't be transcribed. Transcription is not possible. This is only for packaging. But if it loosely packed, transcription is possible, right? So this affects the functioning of DNA, and that is why different levels of compaction are required in the different cell stages. Okay, so now let's learn about the different levels of the compactions in the DNA. Okay, let's have a look at it in detail. Okay, it is here. So very first you have DNA, and you wrap it around the histone proteins, and you form nucleosome. So I'll be discussing about nucleosome in detail in the next video lecture. So the nucleosome, you can also call it as the basic structural unit of chromatin. Okay. Now imagine the DNA. This DNA is a thread and this histone is a button. Okay. Histones protein is button. It's actually wrapped around octamer. Okay, let's this is not matter of concern right now. So imagine histone protein, octamer is like a button and DNA is like a thread. Now you will wind this thread, you will wind this, like you will wrap this thread around this button, and then you'll wrap it around one or two three loops. Okay, then you'll take next button, you'll wrap it around this, and you'll take next button, you'll wrap it around this, and thus you will form a bits on the string structure, which is called as chromatin. Okay, these nucleosomes are arranged consecutively, and they forms bits on the string structure. So DNA is the dimensions of DNA. Okay, the dimensions of DNA are around 2 nanometer, right? But when we pack it into the chromatin, into nucleosome, into the consecutive nucleosome, it gets around 11 nanometer. So see, the dimension is changing. The DNA is getting more compact. Okay. So now you have bits on the string. Now what do? You start coiling this in such way. You start coiling this in such way. Okay, let me just change the color. Let's take it to the blue. Okay. So now you start coiling this in such a way. Okay, so that you put one nucleosome next nucleosome to each other closer, then the next chromosome to the closer to the first chromosome, and next chromosome closer to the previous chromosome. So nucleosome. So this uh, packaging of nucleosomes makes 30 nanometer fiber. It makes 30 nanometer fiber. See, now the level of compaction has been increased, and when you pack this 30 nanometer fiber more densely, you will find the chromosome extensions. So you can imagine it that chromosome is very compact structure. Now you loosen it up, you will get something like this. Okay, now you loosen it up again, you'll get like this. So the extended chromosome, this is extended chromosome, is about the dimension 100 nanometer. Okay, then after this again you coil it, you wrap it intensively, you'll have condensed section of chromosome. Now this is the chromosome, and it is around 700 nanometer. Oh, I made a mistake here. The extended chromosome section is 300 nanometers. Okay, and the condensed section of chromosome is 700 nanometer. Now this is not again enough. You have to pack it extensively, and then you form mitotic chromosomes, the chromosomes which can be seen at the metaphase, which seems very nice bundled structure. This nice bundled structure chromosome, it is of dimension 400 nanometer, and this is the highest degree of compaction that can be achieved by DNA. And now this multiple chromosomes can be fit into the cell nucleus, and the packaging job is done. Okay, I hope this was clear to you. So now the chromosome and chromatin are two different degrees of chromosome and chromatin are two different degrees of compaction. So let's have a look at it. This is how chromosome looks. You can see how extensively it is packed. You can see how extensively it is packed. Okay, you can see it. So these are the arms of the chromosome. Okay, this is the centromeric region. These are the telomeric regions, and this is called distal arm. Okay, so this is chromosome. In this chromosome, when you zoom in, you can see the DNA wrapped around the histones. So this is histone octamer. This is histone octamer. This is histone octamer. This is histone octamer. Around it, you can see how nicely DNA is wrapped. Okay, so the DNA is wrapped around the histones like this. So many times chromosome and chromatin, those two things are confused. Those two terms are very confusing for students. So I have listed down the differences between them. Chromosome is densely packed structure, while chromatin is unwound DNA, which can be found in interphase, and this is found in mitotic phase or meiotic phase. Okay, means cell division phase. It's written here. See, during cell division you can find a chromosome, while during the whole cell cycle, because during whole cell cycle it is metabolically active, right? So you need to transcribe the DNA. That's why it is unwound, so that it is easily accessible. Okay, so the DNA can be used for the macromolecule synthesis, meant it can be used for RNA synthesis in the chromosome, while in the chromatin it can be used for macromolecule synthesis. So this was the differences between chromosome and chromatin. So here I would like to discuss one more thing, that 30 nanometer fiber is a very important stage of packaging, because at the chromatin stage it is loosely packed DNA and you can access it uh, for the transcription. At 30 nanometer fiber, now see, this is DNA as a string. You wrap it around the button. Okay, so this is the button. You wrap the DNA around it 2 to 3 times and then you move on. You took the next button, you wrap it around, you moved on. You took the next button, you wrap it around and you moved on. Now this beads on the string, assume it as it like a necklace which has a lot of beads on it. Okay, and now you have to wind this necklace. You have to wind this necklace to a further degree of compaction. So the scientists, researchers gave two types of models for this binding. Okay, at 30 nanometer level of folding, they gave two models. Okay, those two models differed in topology of the DNA, dimension of the DNA, and nucleosome packaging density. So those two models were one start helix model and two start helix model. So the one start helix model is the model which has packaging density of 11 nucleosomes per 11 nanometer. So beads of string had 11 nanometer dimension. So it accommodates 11 nucleosome in 11 nanometer of the 30 nanometer fiber. Okay, so this is the packaging density. Well, the two start helix model have packaging density lower. Now let me show you diagram in the diagram. Okay, so what happens in one start helix model, the consecutive nucleosomes. This is the nucleosome 1, this is nucleosome 2, and this is nucleosome 3, and DNA is wrapped around this like this in certain number of turns. Okay, so the DNA is wrapped around the nucleosome in such manner. So what do? You start taking this button and you put them next to each other. Okay, like imagine you are stacking the buttons, and the thread around it is also getting closer. Okay, the thread is DNA while the buttons are the histonoctomers. So now you are stacking up the buttons. So now you can have two types of stacking. One is that you directly put them consecutively. You took one button, you took one button. So this is 3D one button. Okay, and you put the other button over it, on the top of it. Okay, you put the third one, you put the fourth one. Okay, so you are just stacking it. So this you see it in the diagram. This is the first nucleosome. Okay, let me change it to the laser so that you can see it better. So this is the first nucleosome, this is the second nucleosome. Okay, and now you can see the third nucleosome here. Okay, I hope that's visible. And then you can see the fourth nucleosome here in the previous. So it is getting stacked in the spiral manner. One, this is two, this is three, and the fourth one is behind. I think it is barely seen, and it is then wrapped spirally like this. Okay, imagine it's a string. Okay, imagine it is a string and you are spiraling the string. Okay, so you are spiraling the string. Okay, I know this diagram is not that good, but just try to imagine it. Okay, yeah, I hope now it's clear. So this is the nucleosome 1, this is the nucleosome 2, and this is the nucleosome 3, and here here you can see the nucleosome 4. So these nucleosomes are then arranged next to each other, and this is what form one start helix model. Okay, this one start helix model is very compact. It has high packaging density. And what was the packaging density? It was 11 nanometer, 11 nucleosomes per 11 nanometer. Okay, this was the packaging density of this one start helix model. Well, the two start helix model is somewhat different. You take the first nucleosome, okay, and then you put the next nucleosome just besides it. But what you do, that you put the third nucleosome besides the second nucleosome but in the opposite direction, and you keep continuing fourth in the opposite direction of the second one, and then fifth in the opposite direction of the third one. Okay, so it is kind of zigzag. Okay, can you see that zigzag thing? Okay, the first, second, third, fourth, and it 1 2 3 4. It's like this: 1 2 3 4. Okay, so it is wrapped up in the zigzag manner. So this is also the two start helix model, is also known as zigzag model. Okay, the zigzag model has lower packaging density. The packaging density is almost 5 to 6 nucleosome per 11 nanometer. Okay, this is the packaging density of the two start helix model. One significant difference is that when you put consecutive nucleosome right next to each other, the linker DNA it gets bent. See here, you can see. I know it's a little bit tough to imagine, but when you see, when you are seeing this, it is getting bent. Okay, it is getting bent. That's why the linker DNA gets bent in a one start helix model. While in the two start helix model, see that this was the linker DNA. It gets here out of the octamer and it goes straight up to the next nucleosome. Okay, so the linker DNA, okay, sorry, linker DNA is straight in the two start helix model. So let's have a quick recap. One start helix model, nucleosomes are adjacent to each other. Packaging density is 11 nucleosome per 11 nanometer. The diameter, the dimensions of this architecture is 34 nanometer. While the linker DNA gets bent. Okay, the linker DNA gets bent. Well, in the two start helix model, it is the zigzag arrangement of nucleosome that stack on the top of each other. See, oh sorry, you can see it's. Wait, it goes like this in the zigzag manner. So on this, they stack on top of one another in the zigzag manner. The packaging density is lower compared to one start helix model. It is 5 to 6 nucleosome per 11 nanometer. The dimensions of this architecture are 24 to 25 nanometer, and the linker DNA stays spread. Okay, so that's it for today. In the next video lecture, we'll be discussing about nucleosomes. Okay, nucleosomes, which are basic structural unit of chromosome. So I already told you that a DNA is wrapped up around histones, and that makes nucleosome. Okay, so till then, see you. Bye bye.